morning, good afternoon, and good evening to everyone who joins us from around the world. We're so glad you're here.
Thank you so much for having me.
This is Andrew and Renee. Welcome to the Stack series.
It's really an honor to be sitting down with you today.
Today, we are exploring the future of mRNA therapeutics, beyond vaccines.
And, I have to say, looking at the source materials for this exploration today, it really feels like we are looking at a map of a territory we haven't even really stepped foot on yet.
I get that completely. I think for most of us, and I definitely include myself in this, our relationship with mRNA kind of started and ended in 2020 or 2021.
Right, the pandemic era?
Yeah, exactly. It was the pandemic shot. It was the fire extinguisher we used to put out a global fire.
That is a really great way to look at it.
We learned the acronym, we got the jab, and we sort of just moved on. But reading through the research, I realized that the COVID vaccine wasn't the destination at all.
Not even close.
It was barely even the starting line. It was more like a proof of concept demonstration.
That is exactly the right way to frame it. The pandemic forced the world to fast-track a technology that had been simmering in the background for, decades, really.
Right.
But now that the infrastructure is built and the safety profile is proven on a global scale, the field is completely pivoting.
So we are moving away from just viruses.
We are moving from a defensive posture, where we are trying to stop an external virus, to an offensive one. We are looking at using this technology to actively treat cancer, to reverse autoimmune diseases, and to fix genetic errors at their source.
It feels like a massive shift in the fundamental philosophy of medicine itself. And I want to get into that comparison early on because the sources keep using this specific analogy of hardware versus software.
Yes, that analogy is everywhere in the literature right now. And it makes perfect sense if you look at the history of medicine. For the last 100 years or so, we have been dealing almost entirely in hardware.
Hardware meaning like pills.
Pills, mostly. Small molecules. Your aspirin, statins, antibiotics, these are chemical compounds.
Right.
You swallow them, they dissolve, and they float around your body until they physically bump into a protein and either block it or activate it.
It's just chemistry.
Exactly. It's brute force chemistry. Then, about 30 years ago, we moved into a new era called biologics.
And these are the monoclonal antibodies, right? The drugs that usually end in MAB, like for arthritis or certain cancers.
You got it. These are large, highly complex proteins, but they are still, in a structural sense, hardware.
How so?
Well, we manufacture the protein in a giant factory, we put it in a glass vial, and we inject it into you. We're giving the body the finished product. To use a classic metaphor, we're giving the body a fish.
And mRNA is different because we aren't giving the fish.
We aren't giving the fish.
Yeah.
We aren't even giving the fishing rod. We are giving the instructions to build the fishing rod.
The blueprints.
We are delivering the blueprints, exactly. We are treating the human body not as a broken machine to be repaired with spare parts, but as a manufacturing plant that just needs a new piece of software to run a different program.
That distinction between the physical drug and the instruction manual is exactly where I want to start our first section today. The foundations.
What we think we know.
Right, because I think a lot of people tuning in have a fuzzy idea of how this actually works. We know it involves genetic code, but how does that actually translate into a therapy?
To really grasp that, we kind of have to go back to high school biology for a minute. We need to look at what biologists call the central dogma.
The central dogma. That sounds almost, I don't know, religious.
In molecular biology, it essentially is. The dogma states that genetic information flows in one very specific direction. DNA makes RNA, and RNA makes protein.
Yeah.
Your body is doing this trillions of times a day as we speak.
Okay, let's break that chain down so we are all on the same page. The DNA is the vault, right?
Right. DNA is the master archive. It sits securely inside the nucleus of the cell.
So it's locked away.
It is highly protected. It is precious. It is permanent. The DNA never leaves the vault. But the cell needs to build things to survive. It needs proteins, enzymes, structural components.
So if the master blueprint can't leave the vault, how does the construction crew know what to build?
That is exactly the problem the cell solves with RNA. When the cell needs to build a specific protein, it goes into the vault, finds the specific page in the DNA book, and makes a photocopy.
And that photocopy is the mRNA.
Exactly. Messenger RNA. The M literally stands for messenger. It is a temporary, disposable copy of the instructions.
Okay, so the messenger RNA leaves the nucleus.
It leaves the vault and goes out into the main workspace of the cell, which is called the cytoplasm. There, it finds a molecular machine called a ribosome.
The sources refer to the ribosome as a 3D printer.
That is the perfect way to visualize it. The ribosome is a cellular 3D printer. It reads the code on the mRNA strand and stitches together amino acids one by one to build the final protein.
And what happens to the instructions once the protein is finished?
Once the protein is built, the mRNA photocopy is shredded by the cell, and the raw materials are recycled. It's totally transient.
So wait, traditional gene therapy, the kind that makes people nervous because it alters your genetics, that tries to change the DNA in the vault.
Correct. Traditional gene editing, like CRISPR, tries to rewrite the master book.
Which is permanent.
It is permanent. It is high risk, high reward. But mRNA therapeutics are fundamentally different. We aren't touching the vault at all.
We are just, what, sliding a fake photocopy into the pile waiting for the printer.
Yes, we are slipping a new work order into the cellular inbox. We are tricking the ribosome into building a therapeutic protein that the body wouldn't normally build on its own.
Like the spike protein for the COVID vaccine.
Right, to train the immune system. Or maybe, and this is where the field is going, it is a protein that acts directly as a life-saving drug.
This brings up the manufacturing side, which blew my mind. I was reading the source materials from Sativa, and it pointed out a massive logistical advantage to this software approach.
Oh, the manufacturing difference is night and day.
Because if we look at those monoclonal antibodies you mentioned earlier, making those sounds like an absolute nightmare.
It is incredibly difficult and expensive. To make a biologic drug, a monoclonal antibody, you have to grow living cells, usually Chinese hamster ovary cells, in these massive, multi-story steel tanks called bioreactors.
You literally have to farm cells.
You are farming microscopic livestock. You have to feed them special broths, keep them at the absolute perfect temperature, monitor the oxygen, and make sure not a single stray bacteria gets in to infect the batch.
It sounds incredibly fragile.
It is very messy biology. It takes months just to yield a small batch of the protein.
And what if the virus you are trying to treat mutates, or if you want to tweak the drug slightly?
You basically have to start from scratch. You have to engineer an entirely new cell line. It is slow, and it is painfully expensive.
But mRNA is different. The sources say it is synthesized via in vitro transcription, IVT.
Yes, IVT. It means you don't need living cells at all.
So no giant steel tanks.
No bioreactors. It is just chemistry in a test tube, or rather a specialized machine. You type the exact genetic sequence you want into a computer using the letters A, C, G, and U.
The genetic alphabet.
Exactly. You add the raw chemical materials, the enzymes and nucleotides, and the machine literally prints the RNA strand.
Just like printing a document.
Just like that. If the virus mutates, or if you want to target a completely different form of cancer, you don't build a new factory. You don't engineer new cells.
You just change the code you typed into the computer.
You just update the software. That is the plug-and-play aspect that has everyone so excited. The manufacturing footprint is tiny compared to a biologics plant.
But there is a very big catch here, isn't there?
There is always a catch in biology.
The catch is that mRNA is incredibly fragile. It is a large, bulky, unstable molecule. And from what I read, your body absolutely hates it.
Loathes it. We have spent millions of years of evolution building up defenses against loose genetic material. Because usually, if there is loose RNA floating around in your blood, it means a virus has breached the perimeter and is actively attacking.
So if you just injected naked mRNA directly into my arm, what would happen?
Nothing therapeutic, that's for sure. Wouldn't work at all.
Why not?
Two major biological roadblocks. First, your bloodstream is heavily patrolled by enzymes called RNases.
RNases.
Think of them as molecular shredders. Their entire job is to patrol the body looking for loose RNA and destroy it on site. They would tear that therapeutic message apart in a matter of seconds.
Well, the instructions get shredded before they even reach the factory floor.
Instantly. But even if, by some miracle, the mRNA survived the shredders, it faces a second impossible hurdle.
Which is?
The cell membrane. mRNA is a large, negatively charged molecule. The protective outer membrane of your cells is also heavily negatively charged.
Oh, like trying to push two magnets together with the exact same pole?
Precisely. They strongly repel each other. The mRNA literally cannot physically cross the cell membrane on its own.
So it needs a vehicle. It needs some sort of disguise, or like a Trojan horse.
It needs an armored transport. And this brings us to the real hero of the story. The sources explicitly call them the unsung heroes of the mRNA revolution.
The lipid nanoparticle, or LNP.
Without the LNP, there is no mRNA therapy. Full stop. It is the sophisticated envelope that allows the fragile letter to actually be delivered.
I really want to spend some time here because I think the general public just glosses over this. We hear the word nanoparticle and it just sounds like magic sci-fi dust.
It does sound like magic.
But the biochemical engineering required to build these microscopic vehicles is just mind-boggling. The sources list 4 key ingredients that make up an LNP. They call them the Fab Four.
The Fab Four lipids, yes.
Can we walk through these? Because it's clearly not just mixing oil and water together.
It is definitely not salad dressing. It is a highly engineered, dynamic piece of nanotechnology.
Okay, let's look at ingredient number 1, the ionizable cationic lipid. The papers constantly refer to this specific lipid as the MVP of the entire system. Why is that?
This is the component that makes the whole impossible journey possible. Here's the engineering paradox. To get RNA into a cell, you ideally want your delivery vehicle to have a positive charge.
Because the cell membrane is negative, so opposites attract.
Exactly. A positively charged particle will naturally stick to the negatively charged cell. But there is a massive problem.
Which is?
If you inject something that is permanently positively charged directly into the human bloodstream, it is highly toxic.
Toxic how?
It rips apart red blood cells. It causes massive systemic inflammation. The body rejects it violently.
So you need it to be positively charged to enter the cell, but neutral to travel safely through the blood.
You need a chemical shapeshifter. And that is exactly what the ionizable lipid is.
How does it shapeshift?
It reacts to its environment. When the LNP is sitting in the glass vial, and when it is injected and floating in your bloodstream at a normal neutral pH of around 7.4, the lipid has absolutely no charge. It's totally neutral.
So it's stealthy. It's just cruising through the blood without causing alarms.
Right. It safely navigates the bloodstream until it bumps into a target cell. The cell sees this neutral particle and decides to swallow it.
Just engulfs it.
This biological process is called endocytosis. The cell wraps the nanoparticle in a bubble made of its own outer membrane and pulls it inside. This bubble is called an endosome.
Okay, so now the Trojan horse is inside the castle walls, but it is trapped inside a bubble.
Yes. And the cell isn't stupid. It wants to digest whatever it just ate to see if it's food or a threat. So the cell starts actively pumping acid into that endosome bubble.
The pH starts dropping.
The environment inside the bubble becomes highly acidic.
And this is the trigger.
This is the magic moment of the entire therapy. The ionizable lipid detects that sudden drop in pH. That acidity chemically forces it to change its structure. It becomes protonated.
Meaning it switches its charge.
It flips the switch from neutral to highly positive.
Oh, wow.
So suddenly, inside this tiny acidic cellular bubble, you have thousands of lipid molecules instantly turning positively charged.
And because they are all positive now, they start repelling each other.
They strongly repel each other, and they are powerfully attracted to the negatively charged inner wall of the bubble trapping them.
So they just tear it apart.
They violently disrupt the membrane. The LNP literally bursts the bubble from the inside out.
That is wild. It's like a chemical depth charge that is perfectly timed to only go off when it is securely inside the target.
That is a brilliant analogy. That process is called endosomal escape. It safely drops the delicate mRNA payload directly into the cytoplasm, right where the 3D printed ribosomes are waiting.
Because without that pH-sensitive switch, the mRNA would just stay hopelessly trapped in the bubble until the cell's acid digested it entirely.
Precisely. It would be a complete failure.
Okay, that is an incredible piece of engineering. That's the MVP engine. But a car needs more than just an engine. What are the other 3 ingredients doing? We have cholesterol.
Right. We usually only think of cholesterol as the bad guy clogging our arteries, but in cellular biology, it is a crucial structural component.
It provides stability.
It is rigid. Right. Inside the LNP, cholesterol acts exactly like the steel rebar in concrete. It physically sits in the microscopic gaps between the other lipid molecules and holds the whole sphere together.
So without the cholesterol, the LNP might just fall apart in the blood.
It would be far too floppy. It might leak the mRNA out before it ever reaches the destination. The cholesterol locks the structure in place.
Okay, structural support makes sense. Next up on the list are the helper lipids.
These are usually standard phospholipids. They naturally mimic the composition of your own cell membranes.
So they act as camouflage.
Camouflage, yes, but they also significantly aid in that fusion and escape process we just talked about. They make the artificial nanoparticle look and behave more like a natural piece of biology, so the cell is more willing to interact with it.
And finally, the 4th ingredient, the PEG lipid, polyethylene glycol.
Ah, the stealth cloak.
Why exactly does this microscopic vehicle need a cloak?
Because your immune system is incredibly paranoid, and rightfully so. If you inject a foreign blob of synthetic fat into your bloodstream, specialized immune cells called macrophages will detect it.
Macrophages being the garbage trucks of the blood.
Exactly. They patrol the blood, and if they see a foreign particle, they will swarm it and eat it immediately. They will clear your expensive drug out of the body before it ever reaches the intended tissue.
So the PEG lipid stops the garbage trucks.
PEG is a very long, stringy polymer molecule that naturally attracts water.
Okay.
So when it's on the outside of the nanoparticle, it pulls in a thick shell of water molecules, creating what we call a hydration layer.
A water shield.
It literally makes the particle physically slippery. It prevents the LNPs from clumping together into dangerous blockages, and it makes it incredibly difficult for the immune cells to get a grip on them.
It buys the particle enough time to circulate and find its target.
Right. But I saw you looking at the notes on the PEG dilemma.
Yes, the PEG dilemma. What is the problem if the cloak works so well?
The dilemma is that the very thing that protects you during the journey eventually becomes a massive hindrance at the destination.
Because if the cloak is too slippery, it can't enter the cell.
Exactly. It's like wrapping a beautiful present in 20 layers of thick duct tape and bubble wrap. It survives the mail perfectly, but the recipient can't actually open the gift.
Oh, I see.
You absolutely need the PEG to stay firmly attached during the perilous journey through the bloodstream, but you need it to magically fall off or shed the exact second it reaches the target cell wall.
So the ionizable lipid can actually touch the membrane and do its job.
Right, so it's essentially a timed release disguise. Tuning that shedding rate is one of the most frustrating and difficult parts of nanoparticle engineering right now.
Okay, so we have laid out the foundations. We have the software, the mRNA, and we have the highly engineered fat bubble, the LNP, which brings us to our second major section, the active debate.
Where the science gets messy.
Because while this delivery system works phenomenally well for a local vaccine injection in the arm, when we try to use it for complex systemic therapies, we run into some serious brick walls.
We do. And the single biggest wall is what the scientific community calls the liver trap.
The liver trap. It sounds like a bad 80s action movie. What is the trap?
It is a quirk of simple human biology that creates a staggering engineering headache. When you inject an LNP intravenously right into the vein, where do you naturally assume it goes?
Well, it's in the bloodstream, so I would assume it just goes everywhere equally.
It does circulate, yes.
Yeah.
But almost immediately, the vast majority of those nanoparticles start accumulating heavily in the liver.
Wait, why the liver? Is the liver just acting as a giant blood filter?
Sort of, but it is actually much more specific than just physical filtering. It all comes back to that stealth cloak we were just discussing.
The PEG layer.
Right. It turns out the cloak is not perfect. As soon as the synthetic LNP enters the chaotic environment of the blood, it immediately gets bombarded and covered in natural blood proteins. We call this phenomenon the protein corona.
Like a crown of proteins around the particle.
Like a crown. Or imagine a thick swarm of bees covering a tennis ball.
Okay, I can picture that.
The particle gets totally plastered. And one very specific protein that absolutely loves to stick to lipid nanoparticles is called apolipoprotein E, or ApoE for short.
ApoE.
Think of the ApoE protein as a highly exclusive VIP pass for the liver. The cells in your liver have special receptors on their surface that are specifically designed to grab anything carrying an ApoE protein.
Oh, wow. So the nanoparticle enters the blood, accidentally gets this ApoE sticker slapped onto it by the body.
And the liver sees that VIP sticker floating by and says, "Right this way, sir." And it aggressively pulls the particle out of the blood circulation and absorbs it.
Which I imagine is actually fantastic if you are trying to treat a rare liver disease.
Oh, it is phenomenal for liver diseases. It is a completely natural, built-in homing mechanism. But think about the broader implications. What if you are trying to deliver a therapy to a glioblastoma in the brain?
Or a tumor in the lung.
Or trying to repair heart tissue after a heart attack?
Then the liver is essentially stealing all of your highly expensive medicine.
Exactly. The liver acts as a massive biological black hole. You inject the drug into the arm, the liver greedily eats 90% of it, and the desperate tumor in the lung gets practically nothing.
That is incredibly frustrating. This is what the sources refer to as the challenge of biodistribution. So how are researchers trying to bypass this black hole?
There are a few major strategies, but one of the most exciting is something called SORT technology.
SORT, Selective Organ Targeting.
Right. It is a really brilliant bit of chemical hacking that was discovered somewhat recently. Researchers figured out that if they simply added a fifth specific lipid to that Fab4 mixture.
Just one new ingredient.
Just one extra lipid, a sort-me molecule. They found they could fundamentally change where the particle naturally ends up in the body.
How does adding one single ingredient change the entire postal code of the drug?
Because it fundamentally alters the internal electrical charge profile of the entire nanoparticle. It turns out the body naturally sorts different particles into different organs based on their subtle charge. By tweaking the chemistry to be slightly more positive, they could suddenly direct the LNPs to bypass the liver and land perfectly in the lungs.
And if they tweak the charge a different way?
They can send them straight to the spleen, which is essentially the headquarters of the immune system.
So they're literally hacking the body's internal mail delivery system.
They are. It is incredibly elegant. But there is another, perhaps more obvious approach being debated, which is active targeting.
Active targeting. This is where you try to attach a very specific physical address label to the outside of the nanoparticle, right?
Exactly. Let's say your goal is to exclusively deliver the mRNA into a specific type of immune cell, like a T cell.
Okay.
You chemically attach an antibody or a tiny targeting molecule called a ligand directly to the outer surface of your LNP, a molecule that only physically sticks to receptors found on T cells.
That seems entirely logical. It's like putting a Deliver to Andrew sticker clearly on the outside of the box.
It is perfectly logical on a whiteboard in a lab.
Yeah.
But remember the biological reality we just talked about, the protein corona.
The swarm of bees.
Right. The massive debate raging in the field right now is whether those expensive targeting stickers actually work in a living human.
Because the blood proteins just cover them up.
That is the major fear. Many scientists argue that the literal second you put that beautifully targeted particle into the bloodstream, the thick corona of blood proteins instantly paste over your targeting ligand.
Like wrapping the addressed package in the thick layers of duct tape.
Exactly. The local post office, the cell, can't read the label anymore because it's completely buried under junk.
So companies are spending millions, maybe billions, designing these hyper-specific targeting stickers, and human blood might just be instantly neutralizing them.
That is a very real possibility. The clinical data so far is extremely mixed. Some early studies show active targeting significantly improves delivery, while other studies show it makes absolutely zero difference in a live animal model. It is a very active, highly contested area of friction in the research.
Fascinating. And another major friction point the sources highlight is immunogenicity.
Ah, yes, the double-edged sword.
This one requires some nuance, because for a vaccine, like the ones we got for COVID, we actively want the immune system to notice the injection, correct?
Absolutely.
Yeah.
For any vaccine, acute inflammation is actually your best friend. You inject the LNP, and you want the local tissue to scream, hey, there's a dangerous intruder here.
That sets off the alarms and wakes up the immune system.
Exactly. It recruits the T cells and B cells to the site so they can study the mRNA protein and build long-lasting antibodies.
Mm-hmm.
In this case, the synthetic lipid nanoparticle itself naturally acts as an adjuvant.
An adjuvant meaning a chemical booster that amplifies the immune response.
Yes. The body hates the lipid, gets angry, and that anger makes the vaccine work better.
But, and here's the huge pivot, what if I am not taking a vaccine? What if I have a severe chronic disease, like I am missing a vital metabolic enzyme, and I need a systemic mRNA injection every single week for the rest of my life?
Then that exact same inflammation becomes your absolute worst enemy.
Because I would just feel sick all the time.
You feel terrible. You do not want a patient developing a high fever, severe body aches, and flu-like symptoms every single Tuesday when they take their life-saving medicine.
No, of course not.
But it gets much worse than just feeling sick. If the immune system keeps getting angry at the delivery vehicle week after week, you eventually trigger the ABC phenomenon.
ABC, accelerated blood clearance.
Right. Basically, the first time you inject the chronic therapy, it works beautifully. The liver makes the missing enzyme. The patient feels great. The second time you inject it a week later, it works, but maybe slightly less effectively.
Because the body is getting suspicious.
Exactly. By the third or fourth weekly dose, the patient's adaptive immune system has definitively learned to recognize the synthetic PEG cloak, or the specific ionizable lipid, as a hostile foreign invader.
It learns the face of the delivery truck.
It creates specialized antibodies, specifically designed to hunt and destroy the truck itself. So on week 5, as soon as that expensive therapy enters the vein, the immune system violently swarms it and completely shreds it before it can deliver even a fraction of the cargo.
The body learns to shoot the messenger.
It flawlessly shoots the messenger.
Yeah.
And for any long-term chronic disease therapy, that rapid clearance is an absolute deal breaker. The drug becomes entirely useless.
So how are the bioengineers trying to solve that?
It is a massive race right now to invent next-generation, ultra-stealthy lipids. They are designing fully biodegradable lipids that do their job and then literally vanish into harmless byproducts within hours, giving the immune system no time to memorize their shape.
That is incredible, just breaking down before the guards can take a picture.
Exactly. Or they're trying to find entirely new classes of lipids that simply do not trip those specific cellular alarm wires in the first place.
Okay, there is one last major scientific debate we need to cover before we move on to the actual clinical applications. The shape of the RNA molecule itself.
Linear versus circular RNA.
Yes, the structural debate. The source is called circular RNA, the Ouroboros.
The mythical snake eating its own tail. It is a very fitting metaphor.
Why does the physical shape of the microscopic string matter so much?
Well, historically, almost all manufactured mRNA is linear. It is a straight microscopic string of code with a defined beginning, a middle, and a distinct endpoint.
Okay.
But inside the cytoplasm of your cells, there are natural housekeeping enzymes called exonucleases.
More shredders.
But these specific shredders act exactly like Pac-Man. They can't just bite the middle of a string. They have to physically find a loose, open end of an RNA strand, attach to it, and then start chomping their way down the line.
Chomp, 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 right down the line.
Precisely. That is the exact mechanism of why mRNA is so wonderfully temporary. It gets eaten from the ends inward. But some brilliant bioengineers recently thought, well, what if we just surgically tie the two loose ends of the RNA string together to form a perfect continuous circle?
If it's a closed circle, the cellular Pac-Man can't find a loose end to grab onto.
You can't find a place to start biting. It just bumps off. Because of this simple geometric trick, circular RNA is insanely, unbelievably stable inside the human cell.
How stable are we talking?
A standard linear mRNA message might survive long enough to be read and printed by the ribosome a few hundred times before it finally degrades. A circular RNA message might survive for days or even weeks, being continuously read thousands and thousands of times.
Meaning it produces way, way more of the therapeutic drug per single molecule.
Exactly. Which fundamentally changes the economics in the patient experience. It means you could potentially use a drastically smaller physical dose of the lipid, causing less toxicity, and the patient might only need an injection once a month instead of once a week.
That sounds like a total game changer. So why isn't everything circular right now?
Because forcefully bending and perfectly sealing a delicate, microscopic string of genetic code into a stable loop inside a manufacturing vat is exponentially more difficult and expensive than just printing a straight line.
Always the manufacturing catch.
Always. But it is definitely one of those promising frontiers in the entire field right now.
Which is the perfect transition. We have covered the foundations, the software, and the fat bubbles. We have covered the active debates over the liver trap and the stealth issues. Now, I really want to get to the part that explains why thousands of scientists are spending billions of dollars trying to solve these problems.
This is the real so what of the whole episode.
The frontiers. What might change everything about how we experience healthcare in our lifetimes. And the first major frontier is the one absolutely everyone wants to hear about. Oncology. Cancer.
It is the holy grail of modern medicine.
But we need to be extremely specific here for the listeners because language matters. When we casually say the phrase cancer vaccine out loud, I think most people immediately picture getting a standard shot in the arm as a teenager and just magically never developing cancer for the rest of their lives, like the polio vaccine.
Right, a preventative shield. But that is largely not what this current mRNA technology is doing at all.
We are talking about therapeutic vaccines.
Yes. Highly targeted treatments specifically engineered for people who have already been actively diagnosed with a growing cancer. The tumor is already established, it is actively evading the body, and we are aggressively trying to retrain the patient's own immune system to suddenly see it and destroy it.
The core concept driving this is called personalized neoantigen vaccines. And I have to say, reading the step-by-step clinical process in the source materials honestly felt like reading a science fiction novel.
It really is science fiction brought to life.
Can we walk the listener through the actual patient journey here? Let's say I go to the doctor and I get the worst news possible. I am diagnosed with an aggressive melanoma on my skin.
Okay, step one in this new paradigm. Your oncology surgeon successfully removes the physical tumor from your body, or at least takes a significant tissue biopsy.
We secure a physical piece of the enemy base.
We take a piece of the enemy. We immediately send that fresh tumor tissue to a highly specialized sequencing lab. They run it through next-generation machines, and they sequence the entire DNA genome of that specific tumor. They read every single corrupted letter of its genetic code.
Okay.
At the exact same time, the nurses draw a tube of your normal, healthy blood, and the lab fully sequences your normal, healthy DNA.
And then the computers compare the two blueprints.
Exactly. They run a massive comparative analysis. They are mathematically hunting for the minute differences. They are looking for the very specific genetic mutations that exist exclusively inside the cancer cells, but are completely absent from your healthy tissue.
And these unique mutations are what the sources call neoantigens.
Neoantigens.
Yeah.
Literally meaning new antigens. Antigens that have never existed in your body until this cancer mutated.
These are the unique fingerprints of the criminal.
Better yet, they are high-definition mugshots. Now, a rapidly mutating tumor like melanoma might have hundreds, sometimes thousands, of these unique mutations scattered throughout its code.
Okay, so we have thousands of targets.
But not all of them are biologically useful for a vaccine. Some of those mutated proteins might be physically buried deep inside the architecture of the cancer cell, completely hidden where the patrolling immune system could never possibly see them anyway.
So how on earth do the scientists know which of the thousands of mutations to actually target with the vaccine?
This is where advanced artificial intelligence and predictive algorithms step in. Humans couldn't do this fast enough. We feed all of those thousands of sequence mutations into a sophisticated AI computer model.
And what does the AI do?
The predictive model simulates your specific cellular biology and predicts which of those exact mutations will be the most highly visible to your specific human leukocyte antigen system, your unique immune system.
It grades them.
It scores and ranks every single mutation. It outputs a report that essentially says, okay, out of 1,000 mutations, these top 10 to 34 mutations are the most glaringly obvious targets. These are the best mugshots to hand to the police.
So we have the top 30 digital mugshots isolated on a lab computer. What is the next step?
The lab takes those 30 digital sequences, sends them to the RNA synthesizer machine, and literally prints a single long strand of custom mRNA that physically encodes the instructions to build all 30 of those mutant tumor proteins.
A completely bespoke, one-of-a-kind piece of software written entirely for my specific body.
Exactly. They encapsulate that custom mRNA into a lipid nanoparticle, they rush the vial back to the clinic, and they inject it directly into your arm.
And what actually happens inside my body after that injection?
Your local healthy cells, maybe the muscle cells in your arm or the cells in your nearby lymph nodes, eagerly take up those LNPs. They read the custom software, and they act as the temporary 3D printer.
So my healthy cells start manufacturing tiny, harmless pieces of my own tumor.
Yes. They manufacture those 30 specific pieces of mutated protein, and then they clearly display them on their outer cellular surface for the immune system to inspect.
They hold up the 30 mugshots.
They hold the mugshots high in the air. Your pathing T cells, the soldiers of the immune system, come along, inspect those displayed proteins, and immediately sound the alarm. They say, wait a minute, this specific shape is completely foreign. This is dangerous.
They get activated and furious.
They become highly activated. They structurally memorize the exact face of the enemy. Then they rapidly multiply and swarm out into your entire circulatory system, aggressively hunting for any cell anywhere in the body that matches that exact mugshot.
And when they eventually find the actual hidden tumor cells,
They launch a devastating precision attack. And this is the profound difference. Standard chemotherapy is essentially carpet bombing. It indiscriminately kills almost anything in the body that divides rapidly.
which is why patients lose their hair and the lining of their stomach.
Exactly. Massive collateral damage. But this mRNA approach is deploying a highly trained sniper team that knows the exact face, name, and address of the target they're looking for, leaving the healthy neighbors completely alone.
The early clinical trial data on this is finally starting to be published, and it is honestly staggering. Let's talk about the Moderna and Merck joint trial, the mRNA 4157 study. They utilized this exact personalized process on severe melanoma patients.
They did. And crucially, they combined this custom mRNA vaccine with an existing blockbuster drug called Keytruda.
Keytruda is a checkpoint inhibitor, right? What does that actually mean in this context?
Think of cancer as being very clever. Tumors often secrete chemical signals that actively put the local immune T cells to sleep. They essentially step on the brakes of the immune system.
So the soldiers are there, but they are sedated.
Right. Keytruda is a drug that physically cuts those chemical brakes. It forces the immune system to stay awake. So, if Keytruda cuts the brakes of the car, The custom mRNA vaccine is providing the steering wheel and the exact GPS coordinates.
And what was the actual result of combining the steering wheel with the cut breaks in the trial?
In severe patients with high-risk resected melanoma, the combination of the custom vaccine plus Keytruda reduced the risk of the cancer recurring or the patient dying by 49% compared to patients who only received Keytruda alone.
I just want to pause on that number, a 49% reduction. We aren't talking about a marginal 3 or 4% improvement that statisticians get excited about. We are talking about literally having the death and recurrence rate for a highly lethal cancer.
It is a genuinely massive statistical leap in oncology. It proves the biological concept works in complex humans, not just lab mice. They are now rapidly moving this exact therapy into massive phase 3 trials for non-small cell lung cancer.
That is incredible. But the study that really gave me pause, the one that felt truly historic, was the BioNTech data on pancreatic cancer.
Oh yes, the autogene cevumeran trial.
Because pancreatic cancer is widely known as the absolute nightmare diagnosis. It is so aggressive and it is usually a rapid death sentence.
It sadly is. And the major biological reason it is so lethal is that pancreatic tumors are notoriously cold tumors.
Well, meaning what?
Meaning they visually have almost no immune cells naturally penetrating inside them. The tumor surrounds itself with a very thick, dense physical shell of stroma tissue. The body's immune system basically just ignores it entirely. It acts like it doesn't even exist.
It is entirely invisible to the body.
Invisible and heavily fortified. BioNTech bravely tried their personalized neoantigen mRNA vaccine on 16 patients with resected pancreatic cancer, just to see if they could even get the immune system to notice the disease.
And did it work?
It successfully induced a strong T-cell response in exactly half of those patients. 8 out of 16 patients responded.
Which for pancreatic cancer is already a massive victory.
But here is the absolute ticker that shook the oncology world. In those specific patients who responded to the vaccine, the researchers went back and drew their blood a full 3 years later.
And 3 years is an eternity in pancreatic cancer survival time.
It is a lifetime. When they analyzed that blood 3 years later, they found massive populations of fierce, highly active T cells, specifically trained by that original mRNA vaccine, still actively patrolling the bloodstream.
It successfully created permanent immunological memory.
It created a durable memory. The actual injected lipid drug had vanished within days, but the highly trained army it created was still actively on guard years later.
That is the literal definition of a functional cure. If a single microscopic cancer cell tries to grow back, the snipers are already on the roof waiting for it.
Exactly. It is profoundly hopeful data.
But looking at the logistics of this, these totally personalized, bespoke vaccines take significant time to manufacture, right? The sources say weeks from biopsy to injection.
Yes. Currently, the absolute fastest turnaround time is hovering right around 4 weeks, which is a miracle of modern science, but
But if you have an incredibly aggressive, fast-moving cancer, you simply might not have 4 weeks to sit around waiting for a lab to print your drug.
that is the grim reality and the biggest limitation of the bespoke approach, which is exactly why massive amounts of research funding are also being poured into what we call off-the-shelf mRNA cancer vaccines.
How does an off-the-shelf vaccine work if every tumor is slightly different?
Because while tumors are unique, many of them share very common foundational mutations that drive their initial growth.
Like shared engine parts.
Exactly. For example, there is a very famous, highly common mutation called KRAS, or the p53 tumor suppressor mutation. Millions of patients globally share these exact same errors.
Okay, I see where this is going.
So, if your biopsy shows you happen to have a standard KRAS mutation driving your colon cancer, the doctor doesn't need to order a custom 4-week print job. They could literally just walk over to the clinic refrigerator, pull out a pre-manufactured mRNA vial that specifically targets KRAS, and inject you that exact same afternoon.
So it is slightly less precise than the fully custom mugshot, but it is infinitely faster and radically cheaper to produce at scale.
Exactly. It is the off-the-rack suit versus the custom-tailored suit. Both might save your life, but one is ready today.
Okay, so that is Frontier A oncology. We are brilliantly teaching the immune system to aggressively attack. But Frontier B is the exact opposite concept. And honestly, reading the biology behind this one felt even more difficult to comprehend. Autoimmune diseases.
It's conceptually much harder. The scientists call it a reverse vaccine.
Right, because in cancer therapeutics, we actively want to break tolerance. We want to start a violent biological war.
Correct.
But in devastating autoimmune diseases like multiple sclerosis or lupus or rheumatoid arthritis, the violent war is already raging out of control. The immune system is actively destroying healthy human tissue. We desperately want to stop the war. We want to create lasting peace.
Exactly. We want to permanently induce immunological tolerance. We want to teach the hyperactive immune system to completely ignore a specific protein it currently hates.
But logically, how on earth do you achieve that with a vaccine? If I have multiple sclerosis, my immune system is actively attacking the myelin sheath that protects my nerves.
Correct. That attack causes the severe neurological symptoms and paralysis.
So if a doctor injects me with an mRNA lipid nanoparticle that makes my body produce even more of that exact same myelin protein, won't that just act like throwing a bucket of gasoline onto an already raging fire? Won't it just make the attack much worse?
That is the exact immense danger, and it is why this research took so long to crack. If you do it improperly, you trigger a massive, potentially fatal flare-up or severe anaphylaxis. The entire genius of the reverse vaccine strategy lies entirely in the biological context.
Context, expand on that.
Your immune system is surprisingly sophisticated. It doesn't just blindly look at what specific protein is present. It heavily analyzes the surrounding environment, the context in which that protein appears.
Okay.
If a strange antigen appears alongside massive danger signals like local cell damage, high acidity, or acute inflammation, the immune system logically assumes this is a violent infection and it viciously attacks.
Right.
But if that exact same antigen is presented to the immune system in a perfectly quiet, completely serene, non-inflammatory environment, the immune system makes a different calculation. It assumes, well, there's no damage here, no alarms are going off, so this protein must actually be a normal, safe part of my own body.
So the goal is to deliver the problematic protein, but do it so quietly that the body is tricked into accepting it.
Yes. We use that highly specialized nucleoside-modified mRNA we mentioned earlier, the chemically altered code that absolutely does not trigger any of the cell's internal danger sensors. We have the liver or the spleen cells gently present the targeted antigen, say the myelin protein for MS, but without ringing a single alarm bell.
And who in the immune system is actually inspecting this quiet presentation?
A very specialized class of cells called regulatory T cells, or TREGs for short.
Treg.
Think of TREGs as the seasoned peacekeepers or the diplomats of the immune system. Their entire job is to prevent civil war. When these peacekeeper TREGs inspect an antigen that is presented in a perfectly quiet, non-inflammatory context, they don't order an attack.
What do they do?
They issue a systemic stand-down order. They actively suppress any other immune cells that are trying to fight that specific protein. They basically broadcast a signal saying, everybody calm down, this myelin protein is an ally, do not shoot.
Wow. And the BioNTech study on multiple sclerosis in lab mice actually proved this concept works in reality, right?
It was a stunning piece of literature. They used a well-established mouse model for MS called the EAE model, where the mice progressively suffer severe paralysis. They injected this quiet, tolerance-inducing mRNA.
And it stopped the paralysis?
It rapidly halted the disease progression. The mice regained motor function. But honestly, the most scientifically important part of the data was that the vaccine solely stopped this specific attack on the myelin. The rest of the mouse's immune system remained completely active and totally healthy.
Why is that specific distinction so crucial for patients?
Because the current state-of-the-art pharmaceutical drugs we have for severe MS or lupus are incredibly blunt instruments. They essentially suppress the patient's entire global immune system to stop the symptoms.
Which leaves the patient incredibly vulnerable.
Exactly. Patients on these heavy immunosuppressants can easily end up in the ICU or die from a simple common cold or mild fungal infection.
But this targeted mRNA approach is like a sniper shot for peace.
Exactly. It completely halts the devastating civil war against the nerves, but leaves the entire military perfectly intact and fully armed to fight off real outside invaders like the flu.
There is also this completely fascinating, almost unbelievable application the sources mentioned for type 1 diabetes coming out of research from the University of Chicago. This one uses what they call a cloak strategy.
Oh, this cell self-defense strategy. I absolutely love the elegance of this one. It's brilliant.
Break it down for us, because in type 1 diabetes, the patient's immune system tragically targets and destroys the beta cells in the pancreas, which are the only cells that manufacture insulin.
Right. And without insulin, you cannot regulate your blood sugar, and the disease is ultimately fatal without continuous lifetime injections.
So how does mRNA protect the pancreas?
The researchers approached the problem from a totally lateral direction. They asked a brilliant question. How do severe cancer cells consistently manage to survive and hide from a highly aggressive immune system?
By using chemical disguises.
Exactly. Cancer cells very often cover their outer surface in a specific biological protein called PD-L1.
PD-L1.
Think of PD-L1 as a universal "don't eat me" signal. It acts as an invisibility cloak. When an angry immune cell bumps into a cancer cell covered in PD-L1, the cloak forcibly turns the immune cell off. It makes it walk right past.
So the cancer steals a biological shield to hide in plain sight.
Right. So the Chicago researchers looked at that and said, why don't we purposefully give that exact same invisible shield to the healthy pancreas cells that are currently under attack?
They intentionally gave the good guys the bad guys' ultimate weapon.
Precisely. They engineered an mRNA sequence that carried the instructions to build that PD-L1 shield protein. They delivered it directly into the surviving beta cells in the pancreas.
And the beta cells read the software and printed the shield.
They printed the shield and proudly displayed it on their surface. And it miraculously worked. The autoimmune T cells would rush in to attack the pancreas, hit the PD-L1 shield, and just harmlessly turn off and walk away. The immune system completely stopped attacking, and the diabetic mice successfully continued to naturally produce their own insulin.
It is using the deep rules of the biological game against itself.
It opens up an entirely new world of possibilities. Think about the implications for something like organ transplants.
Oh, wow.
Right now, if you get a life-saving kidney transplant, you have to take harsh anti-rejection drugs every day for the rest of your life so your body doesn't destroy the new organ.
Which ruins your immune system.
But imagine if, just hours before the surgery, we simply infuse the donor kidney with this specific mRNA software, instructing the kidney cells to rapidly produce that PD-L1 invisibility cloak.
We just make the transplanted organ totally invisible to the host's body.
Exactly. You might completely eliminate the need for a lifetime of toxic anti-rejection medications. The body would just accept the foreign organ as its own.
My mind is genuinely blown. Okay, let's move to Frontier C, the drug factory. This concept brilliantly connects right back to that major biological hurdle we discussed earlier, the liver trap.
Yes, the notorious liver trap.
We spent 10 solid minutes earlier bashing the liver for selfishly eating all of our carefully engineered nanoparticles.
We did. It's a massive headache for targeted delivery.
But the sources noted that some highly pragmatic researchers looked at that exact same frustrating problem and said, wait a minute, the human liver is literally the most powerful, efficient protein manufacturing factory on the planet.
It makes almost all the crucial proteins circulating in your blood right now.
So instead of constantly fighting the liver trap, why don't we just use it to our advantage?
Instead of stubbornly swimming against the biological current, just turn around and ride the wave.
Exactly. Let's look at encoded antibodies. Say a patient urgently needs a complex monoclonal antibody treatment to fight off a severe Zika virus infection or chikungunya, or they need a bispecific antibody for an advanced cancer. We already established earlier that physically manufacturing those giant proteins in a commercial facility is a nightmare.
It takes months, requires sterile bioreactors, and costs anywhere from $50 to $100 per bare gram of protein.
Extremely expensive and painfully slow.
So the brilliant pivot is this. Instead of spending millions manufacturing the fragile physical protein in a lab and trying to safely ship it across the world in a freezer, we just sequence the genetic mRNA instructions for that specific antibody.
The software code for the cure.
We package that cheap, simple mRNA code into a standard lipid nanoparticle, and we just inject it into the patient's arm.
And because of the liver trap.
The LNP naturally and efficiently goes straight to the patient's liver. The billions of highly efficient liver cells eagerly absorb the mRNA software, fire up their internal ribosomes, and they rapidly start pumping massive amounts of the life-saving antibody directly into the patient's own bloodstream.
You literally turn the patient's own organs into the pharmaceutical bioreactor.
You completely outsource the heavy manufacturing to the human body itself. It is infinitely cheaper to scale. You entirely remove the complex cold chain storage requirements for transporting fragile proteins to developing nations. You just need to safely transport the stable code.
This precise mechanism could be absolutely massive for treating rare, devastating genetic diseases, too. The literature specifically highlights conditions like propionic acidemia.
Yes, or methylmalonic acidemia. These are truly heartbreaking, devastating metabolic diseases.
Walk us through why mRNA is such a perfect fit for these conditions.
With these rare diseases, a child is unfortunately born missing a single specific working gene in their DNA. That single error means they cannot produce one specific metabolic enzyme. And without that single enzyme, normal digestion fails, and highly toxic metabolic byproducts rapidly build up in their tiny bodies.
And the physical results are catastrophic.
Severe brain damage, recurring comas, massive heart failure. It is tragic.
And right now, what is the standard of care for these kids?
Honestly, there is no real cure. It is extremely strict, grueling dietary management, hoping to minimize the toxins. It is just managing a very slow decline.
But with this new mRNA factory approach, we can completely change that equation.
We can bypass the broken DNA vault entirely. We just continuously deliver the correct, synthesized mRNA code for that exact missing enzyme. The LNP naturally homes right to the liver.
The liver absorbs the correct instructions.
And the liver flawlessly prints the missing enzyme. The metabolic engine suddenly has the exact part it needs, and it runs smoothly. The toxic buildup simply stops.
But because it is mRNA, and we know mRNA is naturally temporary and gets shredded,
Right.
this wouldn't be a one-time permanent genetic cure. They would need continuous maintenance injections. How often are we talking?
Based on the current half-life of the technology, they would probably need a fresh injection every few weeks. Very similar to how a type 1 diabetic needs regular daily insulin injections to survive. It is an ongoing maintenance therapy.
But for a devastating pediatric disease that currently has absolutely zero real treatment options, a shot every 3 weeks to live a completely normal life is nothing short of a miracle.
It is a profound medical miracle. It completely shifts the paradigm of genetic disease management.
This logically brings us right into our final and perhaps most difficult section of the exploration, the stark implications.
The messy reality of economics and society.
Yes, because we have painted a very rosy, highly optimistic, techno-utopian picture here for the last hour, but we absolutely have to ground this in reality. And the biggest, heaviest reality is the sheer cost of this personalization.
The cost is the massive elephant in the room that no one in the shiny press releases really wants to talk about.
Let's talk real numbers. A fully personalized cancer vaccine where we surgically biopsy your specific tumor, rent time on a supercomputer AI to sequence your genome, and physically print a custom, one-of-a-kind drug vial entirely for you. That is not going to be $20 at the local pharmacy.
No. Current industry estimates, factoring in the intensive labor, the complex machinery, and the highly specialized supply chain, place the cost of a single personalized treatment course at easily over $100,000 per patient.
$100,000 just for the manufacturing.
And that price tag might actually be conservative in the early years.
So the immediate glaring question becomes, who on earth actually pays for that? Can modern insurance systems or nationalized health services realistically absorb the cost of a bespoke $100,000 drug for every single cancer patient?
That is the central, agonizing policy debate. In the short term, health systems will absolutely buckle under that sudden financial weight.
But the sources present a very compelling long-term economic counterargument, the curative economic model.
Yes, the long-term math looks very different if you step back. Consider the grim alternative trajectory. A patient diagnosed with aggressive metastatic melanoma might tragically spend 3 to 5 years bouncing in and out of intensive hospital care.
Right.
They will endure multiple traumatic surgeries, rounds of damaging radiation, months of failed, highly toxic chemotherapy, expensive stays in the ICU, and eventually costly palliative hospice care.
Which is a brutal experience for the patient.
A horrific experience. And from a purely cold actuarial economic standpoint, that drawn-out 5-year process can easily cost a healthcare system $500,000 to $1 million.
Wow.
And at the end of all that immense spending, the patient might still tragically pass away.
So if you frame it like that, suddenly spending a flat $100,000 right up front to successfully train the immune system and actually functionally cure the patient in month 1 is effectively a massive financial bargain for the insurance company.
In cold economic terms, yes, it saves massive amounts of money. A highly effective one-time cure is almost always drastically cheaper than years of chronic failing disease management.
But the structural problem is that healthcare budgets aren't designed like that.
Exactly. Health insurance companies and hospital budgets operate on strict 12-month annual fiscal cycles. They do not calculate long-term 10-year lifetime value cycles. Paying $1 million over 5 years is somehow structurally easier for them to absorb than paying $100,000 on a Tuesday. It requires a fundamental systemic shift in how we fundamentally price and define medical value.
And while we wait for the lumbering insurance bureaucracy to figure out that math, what happens to patient access? Is this incredible life-saving technology merely going to be reserved exclusively for ultra-wealthy individuals who happen to live near major research hospitals in Boston or London or Zurich?
That is a very real, very terrifying risk. The initial rollout will almost certainly be highly unequal.
But there is a technical aspect to mRNA manufacturing that might naturally counter that inequality, right? The physical footprint of the technology itself.
Yes, the technology fundamentally possesses a highly democratizing feature. Remember earlier when we compared the massive multi-billion-dollar steel bioreactor plants needed to make traditional antibodies?
Right, the giant cellular farms. You can't just easily build one of those in a remote rural village.
Exactly. But to manufacture clinical-grade mRNA, you do not need a billion-dollar factory. You just need a specialized, sterile chemical synthesizer.
And physically, how big is that machine? Like the size of a microwave?
Maybe the size of a standard kitchen refrigerator, once you include the purification pumps. But it is relatively very small. This has led to the incredibly exciting concept of the pop-up lab.
Pop-up GMP manufacturing.
Exactly. There's a very famous, highly ambitious pilot project currently being deployed by BioNTech called the BioNtainer.
The BioNTech.
It is essentially an entire, fully functioning, hyper-sterile GMP pharmaceutical manufacturing laboratory, brilliantly crammed into 6 standard interlocking shipping containers.
A literal state-of-the-art lab in a box.
A lab in a box. You can physically load these containers onto a cargo plane and drop them directly into Kigali, Rwanda, or rural Brazil, or a remote province in India.
And how do they know what drug to actually manufacture once they're in the jungle?
They don't need to ship frozen, fragile glass vials from a factory in Belgium anymore. The doctors in the remote clinic just diagnose the local circulating virus or sequence the patient's tumor, and the central headquarters simply uploads the digital genetic code to the BioNtainer via a secure satellite internet connection.
They literally email the life-saving medicine.
They email the file, and the machine locally prints the physical vaccine right there on the ground, hours away from the patient.
That completely fundamentally upends the entire global pharmaceutical supply chain.
It creates a radically decentralized, highly resilient manufacturing model. It turns physical medicine into pure digital information. Once you have the 3D printer installed, you just need access to the open source file.
Information medicine. That really is the perfect overarching theme of this entire discussion. We are actively moving from the heavy industrial age of medicine, building these crude, heavy chemical machineries, directly into the sleek information age.
We truly are, and honestly, just like the wild, unpredictable early days of the internet in the 90s, we are only just barely beginning to figure out what this biological network can actually do.
It's like we successfully started with basic email, which was the COVID vaccines.
Right. Simple, highly effective message transmission.
And now we are rapidly figuring out complex e-commerce, which is the personalized cancer treatments and the autoimmune reverse vaccines.
Exactly. And the really crazy part is, who knows what the equivalent of social media or globally integrated cloud computing looks like in this biological analogy 10 years from now.
That is simultaneously a genuinely terrifying and incredibly exciting thought to leave on. Because what happens when biohackers figure out how to write and share their own open source medical software patches on the internet?
The regulatory and ethical nightmare that we'll create is truly unimaginable. But the sheer potential to finally fix the biologically unfixable, to cure the incurable, is finally tangibly within human reach.
It really feels like we are standing together on the sandy shore of an entirely new medical continent. We've safely mapped the beach, but the deep, vast jungle is just waiting to be explored.
And I, for one, absolutely cannot wait to see what incredible things we find in there.
Before we wrap up, thank you to all our partners, sponsors, and scholars who made this podcast and the research possible. We're truly grateful for your support.
And to our listeners, thank you for being part of this journey with us. We hope you enjoyed today's discussion. If you did, be sure to subscribe and support us in Patreon. And of course, you can write to us in comments if a specific topic needed to be covered, which help us all to grow.
Until next time, Stay curious, keep learning, and let the search for wisdom guide you forward. Have a great day.